Mechanisms first. So our speaker today is Dr. James Fagan, who is head of Division of Subspecialty Medicine, Human Oncology and Pathogenesis program at MSKCC, which is Memorial Sloan Kettering Cancer Center. MSK has ranked in the top 2 best hospitals for cancer by U.S. News and World Report every year since 1990. And it's one of the oldest hospitals for cancer centers in U.S. Dr. Fagan's group has been instrumental in characterizing somatic genetic changes associated with thyroid tumor initiation progression and in defining their functional consequences. They have focused on the role of MAP kinase effector on thyroid tumorigenesis and their impact on the differentiated state of thyroid cancers. They defined the active selective kinase inhibitor on thyroid cancer cell growth and responsiveness to radioactive iodine and identified unique linkage-specific mechanisms of adaptive acquired resistance to these drugs. New information on the genomics of advanced thyroid cancers have been provided insights into events associated with disease progression, the consequences of which his team explores in various model systems with the goal of developing new mechanism-based therapies for most virulent forms of the disease. And the panelists today, we have Dr. Ming Zhaoxing, who is Dean of School of Medicine at Southern University of Science and Technology in China. Dr. Michael Ming Zhaoxing is the founding Dean of School of Medicine and Chair Professor at Southern University of Science and Technology in Shenzhen, Guangdong, China, and Professor of Medicine, the Johns Hopkins University School of Medicine in Baltimore, Maryland, USA. Before joining SysTech, he had been tenured Professor of Medicine at Johns Hopkins University School of Medicine. He was also Professor of Oncology, Pathology, and Cellular and Molecular Medicine, Director of the Johns Hopkins Clinical Thyroid Tumor Center, the Chief of the Laboratory of Cellular and Molecular Thyroid Research at the Johns Hopkins University School of Medicine. His team has pioneered the early detection clinical translation of BRAF and TERT promoter mutations in thyroid cancer and has been thus granted 10 U.S. patents. So Dr. Xing has been awarded for many of the honors that you can see here, and he has served as the chairman of the ATA organizing committee and scientific committee, and was a member of the Association of American Physicians. And we're also very honored to have Dr. Jnana Krishnamurthy, who is a senior research scientist at Human Oncology and Pathogenesis program at MSKCC. Dr. Krishnamurthy's research addresses critical knowledge gap that exists in identifying the role of thyroid cancer drivers in tumor development and disease progression with a goal towards uncovering novel therapeutic vulnerabilities. His research has used in vitro and in vivo experimental models to explore the functional consequences of thyroid cancer drivers, including F1AX, RBM10, and members of the SNF complex genes, and they are cooperated with RAS or BRAF mutations in driving advanced thyroid cancers. Recent research efforts have focused on understanding the role of RBM10 loss of function mutation in thyroid cancers and preclinical investigations to improve RAI efficiency in BRAF V600E driven papillary thyroid cancers and develop mechanism-based combination therapies using advanced thyroid cancer models. We are also very honored today to have Dr. Gopal Iyer with us, who is head at the Department of Head and Neck Surgery, National Cancer Center, and Singapore General Hospital. Dr. Iyer graduated top of his medical school class at NUS in 1998 with honors. He subsequently went on to complete a PhD in molecular Carcinogenesis at the University of Cambridge under Dr. Carlos Cades. Trained as a general surgeon at Singapore General Hospital, he went on to 2 separate head and neck surgical fellowships at Sydney Head and Neck Cancer Institute at Royal Prince Alfred Hospital, Australian Memorial Sloan Kettering Cancer Center, New York, USA. At the end of his fellowship at MSK, he was awarded the Michael Bird Award for being the best fellow in surgical division, both in terms of operative skills and research output. We're also very happy today to have our one of our previous guests back with us, Dr. Oi Lin Kong, who had joined us previously, and a lot of our audience may recognize her. Dr. Kong is board certified in internal medicine. She's also an MBBS and a fellow of the Royal College of Physicians. Her research focuses on cell therapy research in metabolic diseases. Next, we have Sun Yaoting, who is a postdoc researcher from Guoming's lab, who was previously a PhD student at the lab. She just graduated this year, actually. And last but not the least, we're very happy to have Dr. Tianan Guo with us. Dr. Guo is a principal investigator at Westlake University. His current research focus on developing proteomic technologies for precision medicine, exploring diagnostic biomarkers for thyroid cancers, and understanding understudied proteins. At the end, before we start our first talk, I just want to briefly let some of our audiences who might be interested in applying for a PhD program. Westlake University is actively enrolling, and you have until March 31st to enroll, and it doesn't matter if you're a bachelor or master. Go ahead and come to Westlake University website and start your admission application process. Now, without further ado, let's please welcome Dr. Fagan to start his talk today. Okay.
Thank you very much. So as soon as you put yours down, I'll share my screen. There we go.
Yes, please. Yes, please.
Well, thank you again for the invitation. It's a real pleasure to be here, actually, and also to see some old friends. Obviously, this is a very broad topic. I wanted to cover various aspects of thyroid cancer pathogenesis, in part because this talk was prompted by a collaboration that we are developing with Dr. Guo's group concerning proteomics of thyroid cancer cell lines. So, as some of you may be aware, a group of us a few years ago did a genomic and transcriptomic characterization of the existing thyroid cancer cell lines. At the time, it was just over 50. Obviously, getting proteomic information on these cell lines would be extremely valuable to the research community. So I thought that I should give you like a bird's eye overview of thyroid cancer pathogenesis, focusing on a few areas that could be of value to the proteomics group as you start thinking about the major questions in this field. So I believe you probably, or many of you, are familiar with the TCGA study of papillary thyroid cancer. This slide is essentially just sort of a summary of the main genetic abnormalities found in this disease. Papillary thyroid cancers are amongst the most genomically simple tumor types, and they're characterized by mutations of effectors in the MAP kinase pathway. And primary among them are mutations of BRAF, primarily the BRAF V600E mutation, much less frequently class 2 BRAF mutations. I'll discuss these briefly later. In addition to this, there are mutations of the 3 RAS genes, with NRAS being the most significant one. And besides this, there are also fusions of primarily receptor tyrosine kinases, as well as of BRAF itself. And as you can see from this oncoprint, These mutations are essentially largely non-overlapping. And the common pathway that they all share is the activation of MAP kinase. And that is why this disease in particular is one of the prototypes of conditions which are associated with constitutive activation of this pathway. These mutations altogether explain roughly about 85% of all thyroid cancers. There's also a subgroup where there is no obvious driver mutation, but these are associated with significant copy number abnormalities, as shown here. Other than this subgroup, from the standpoint of copy number changes, this is a relatively quiet disease. Most of these tumors are diploid. There are exceptions, and I'll talk about that in a minute, with certain recurrent events, such as amplification of chromosome 1q that I will go into later, as well as chromosome 22q deletions as well. Now, this overall genomic simplicity allowed the TCGA to actually correlate the main genetic drivers of the disease with the other integrated platforms that were being analyzed, including RNA-seq, as well as methylation clusters and reverse protein phase array, which was the approach that they used to characterize a selected number of post-translational modifications in key proteins. And as you can see here, what they based on this, what was immediately obvious was that the mRNA clusters seemed to track very closely to whether the disease was associated with RAS mutations or BRAF mutations. And based on this, the TCGA created a 71 gene signature that distinguishes thyroid cancers as either resembling RAS-like tumors, and so we call those RAS-like tumors, and the second group are the ones that resemble BRAF tumors, and those are called BRAF-like tumors, of course. You can see microRNA clusters also segregated quite beautifully with this characterization, as well as the changes in the composition of post-translational changes in these proteins. There's also a strong histological correlation, and I'll get to that in a minute. Now, when you then look at the individual mutations that are either BRAF-like and RAS-like, aside from RAS mutations, you can see that the various fusion proteins lie somewhere in between these two polar changes, with RET rearrangements overall, which is the most common fusion, tending to be more BRAF-like in their gene expression profile. Now, I wanted to focus a little bit on the slide that describes the proteomic changes as defined by RPPA because of the nature of this conversation that we are having today. So BRAF-like tumors, based on the overall signaling proteins, which are shown here, so here in the lower panel on the right side, you can see particular phosphorylation events primarily associated with either BRAF-like tumors or RAS-like tumors. And I want to actually focus on just a few of these. What we do know about the signaling downstream of BRAF is that BRAF, as contrary to what normally happens with RAF proteins, tends to signal as a monomer, whereas normally in cells that are responding to receptor tyrosine kinase ligand associations, which result in activation of RAS, this normally is associated with RAF dimerization. And that RAF dimerization then signals downstream. Once ERK is activated, then there is a negative feedback that actually phosphorylates CRAF on phosphoserine 338, which essentially interrupts this dimer formation and is a negative feedback loop that dampens the output of the pathway. And as you can see quite beautifully, that if you look at the CRAF phosphoserine 338 row here, what you can see is that it is activated, this phosphorylation, in the RAS-like tumors, but not in the BRAF tumors. And that is because BRAF does not require dimerization, and therefore there is no need for CRAF phosphorylation to interrupt its signaling. Because of that, the output of the MAP kinase pathway, because it is unresponsive to negative feedback, is higher. And that is manifest through the expression, and this is RNA expression, of certain downstream effectors of this pathway, which are the dual specificity phosphatases in this example, which are markedly increased in BRAF-like tumors, but not much less so in RAS-like tumors. Now, this is, of course, also a source of negative feedback, which is more distal. So these dual specificity phosphatase, among other targets, dephosphorylate ERK itself. So these are important differences in how these two oncoproteins, RAF and RAF-like proteins and RAS, actually activate the MAP kinase pathway with different intensities in the flux of the pathway. In addition to that, of course, RAS-like tumors signal as well through PI3 kinase. And you can see that quite nicely here through the differences in the activation of downstream substrates of PI3 kinase, the AKT proteins, which basically at their two phosphorylation sites, 308 and 473, are high in RAS-like tumors, but not as high in the BRAF-like tumors. So this cements what had been previously done in terms of biochemical characterization of these oncoproteins, in large part in the lab of Neil Rosen, who characterized the fact that BRAF V600E, which are the class 1 BRAF mutations, signal as monomers. So if we boil all this information down into like an integrated picture of one of the lessons we learned from the TCGA, you can see here on the left panel that BRAF tumors primarily associated with classical papillary thyroid cancers or tall cell variant papillary thyroid cancers. These are predominant histological subtypes associated with this mutation. Altogether, 62% of these tumors are characterized by papillary thyroid cancer. By contrast, another type of differentiated cancer, follicular thyroid cancers, do not normally harbor BRAF V600E mutations. Here again is a simplified cartoon of the way this BRAF mutations signal and the unresponsibility to negative feedback. And the consequence of this is that they have a high ERK transcriptional output. Now, what I did not mention in my previous slide is that the higher the ERK transcriptional output, the more suppressed the thyroid differentiation program is. Whereas in RAS-like tumors, they're primarily associated with follicular variant papillary thyroid cancers. They represent 12% of the total. Also, they are prevalent in follicular thyroid cancers. We've discussed already how they tend to signal. The ERK transcriptional program is actually activated, but relatively dampened as compared to BRAF tumors, and therefore the thyroid differentiation program is less suppressed. Finally, there's other areas in which these two cancers differ in interesting ways. What we know is that the thyroid cell polarization and the structure of the thyroid follicle is dependent, based on the work of Pilar Santisteban's lab in Madrid, on the expression of this lineage transcription factor PAX8, which drives, among other genes, expression of cadherin 16. That's essential for thyroid cells to adopt their polarity between the basolateral and apical membranes and for them to be able to form this circular follicle. This is actually suppressed by the MAP kinase pathway. So in the BRAF-like tumors, since this program is essentially disrupted, they lose their follicular structure. By contrast, in the RAS-like tumors, it is comparatively retained. That is why they're considered follicular variants of papillary thyroid cancer. Now, this relationship between MAP kinase output and differentiation state is not simply an association, it's also causal. And this is from a study published a few years ago in a mouse model in which BRAF V600E is expressed in the thyroid in a doxycycline-inducible manner. In this way, when mice are treated with doxycycline, they actually the uptake of iodine 124 in the thyroid is suppressed. So this is 1 week on doxycycline. And if you allow the doxycycline actually to wash out for 1 week, the uptake of radioactive iodine is restored. If you treat them with just a vehicle, obviously, the restoration does not take place. But if you interrupt the MAP kinase pathway with either a RAF kinase inhibitor or a MEK inhibitor, you can restore iodine incorporation. In this same study, what was shown is that this was associated with restoration of expression of thyroid lineage transcription factors, PAX8 and NKX2.1, as well as expression of the sodium iodide symporter, thyroglobulin, thyroperoxidase, all of the genes required for thyroid hormone biosynthesis. And concerning the I-124 uptake, this is quantified here. This is in the context of wild-type thyroid. 1 week after switching on BRAF, the uptake of iodine goes away. It's restored by washing out doxycycline, and it's also restored by treatment with MEK and RAF inhibitors, like I mentioned. Now, this has also now been translated into patients. So in patients who have metastatic thyroid cancer that is refractory to radioactive iodine, and here you have one patient in panel A on the left, where this patient had an I-124 scan, and iodine 124 was administered to this patient, and the scan was taken, and you can see uptake in the normal physiological sites, the gastrointestinal tract, the bladder as part of the elimination of I-124 in the urine, salivary glands primarily, but none of the lung metastases trap iodine. When you treat this patient with a MEK inhibitor, selumetinib, you actually restore iodine uptake and actually iodine responsiveness in a subset of these patients. I'm not going to get into this any further. There have been a number of clinical trials now that have been published and others that are in progress that are trying to find the right clinical place for this therapeutic strategy. But just to illustrate the point that the activation of the MAP kinase pathway and the suppression of the thyroid differentiation state are not simple associations, but they are causally related to each other. So then what is the biochemical basis or the functional basis for this, association or yin-yang relationship between MAP kinase activation and differentiation state? So what we know is that when you have activation of the MAP kinase pathway, as illustrated here on the left, by a mutation of BRAF, you drive phosphorylation of MEK and ERK. That in turn suppresses the ability of lineage transcription factors such as PAX8 and FOXY1 to access chromatin and to actually activate thyroid differentiation gene expression. In addition to that, they also suppress the actual expression of these lineage transcription factors. So their biosynthesis is decreased, and their ability to bind DNA is also blocked because chromatin becomes more compact. In this setting, of course, the thyroid differentiation program is suppressed. Now, normally, thyroid differentiated function is regulated through TSH, a glycoprotein made by pituitary cells, through binding to the TSH receptor. This in turn activates heterotrimeric G proteins, primarily GS-alpha, to activate adenylyl cyclase, and in turn this drives cyclin-MP, protein kinase A, and this is a strong activator of thyroid differentiation. But in the context of a BRAF mutation, there is impaired expression of the receptor for TSH, and there is also a block on the ability of adenylyl cyclase to actually generate cyclic AMP. So at least at two nodes, activation of the MAP kinase pathway is suppressing the cyclic AMP-driven differentiation program. So I'd like to sort of now change gears a little bit into a recent study that we've completed that hopefully will be published within the next month or so, looking at the exceptional responders to radioactive iodine therapy. So for those that are not clinicians, radioactive iodine has been used to treat patients with thyroid cancer for now more than 60 years. Having said that, it has never undergone a rigorous clinical trial. So radioactive iodine therapy has entered practice before the FDA and other regulatory bodies around the world had a chance to evaluate it in the way a drug would be evaluated today. Most of the use of radioactive iodine is in the adjuvant setting. So we tend to use radioactive iodine in patients after surgery, if they are relatively high risk for recurrence, we give them a dose of radioactive iodine essentially to try and decrease those recurrence rates. But in measuring the efficacy of radioactive iodine therapy, we essentially, have a lot of difficulty distinguishing to what extent the disease has responded to surgery itself or to the adjuvant treatment with radioactive iodine. Radioactive iodine is also used for patients with distant metastatic disease. In this situation, the patients have structurally significant metastasis primarily to the lung and bone. And so you would have thought that there would have been a study looking in a randomized way of whether radioactive iodine or placebo or an alternative therapy would be beneficial. But that study has never essentially been done. So we really don't know or don't have any true measure of how efficacious radioactive iodine is in terms of structural disease control. So my colleague Laura Bucay at Memorial Sloan Kettering decided to look at this in collaboration with our group. And what she did is a retrospective look at all patients who had been treated with radioactive iodine at Memorial Sloan Kettering between 1985 and 2018. And we found altogether that 20,000 patients had been treated, and of those, 5,000 had been treated at Memorial Sloan Kettering, and 3,300 of these had undetectable thyroglobulin antibodies in their blood. And this is important because of the approach that we use to try and identify patients with metastatic disease. If they have autoimmune thyroid disease and therefore they have thyroglobulin antibodies in their blood, the biomarker that we normally use, which is measurement of thyroglobulin, is essentially less informative. So in these 3,300 patients, what we looked for were patients who had a thyroglobulin level at any time greater than 200 nanograms per milliliter after their operation. When you get a level of this, greater than 200, virtually all these patients will have distant metastatic disease. So of these 3,300 patients, only 221 met this criteria. And we then looked at what their nadir, what their lowest thyroglobulin was after treatment with radioactive iodine. And we specifically looked for those patients who had a nadir less than 20. And only 34 met this criteria. And of these, only 11 met what is considered an appropriate oncological criteria for a response, which is based on RECIST version 1.1, which is a reduction in size of greater than 30%. So 11 of these 221 met this criteria. So overall, you can see the rarity of this overall type of response. And so we were ultimately able to consent only 8 patients, and we did a case control. So we matched very carefully non-responders who had essentially identical presentations in terms of the histological type of the cancer, the stage of presentation, and so on. This is the table that actually characterizes that. I'm not going to go to this, the demographics of this in detail, other than to just point out that, as you might expect, the median nadir after thyroglobulin after radioactive iodine was 0.8 in the responders and 187 in the non-responders. And their median time to disease progression was significantly different. And that will be illustrated further in the next slide. So these are just examples. This is a patient with structural metastatic disease as shown here. After treatment with radioactive iodine, these lesions are no longer detectable. This is the patient's body scan. These are just other examples of patients. And what we are calling these as either exceptional responders in red or non-responders in blue. And this is their progression-free survival data. You see very markedly different, as well as disease-specific survival. So we then did a whole exome sequencing, as well as characterize them by the cancer exome panel that we used at Memorial Sloan Kettering, which is called MSK Impact. And these are the main drivers of the exceptional responders versus the non-responders. So as you can see, in the exceptional responders, none of these patients harbored a class 1 BRAF mutation, which is the BRAF V600E or other V600 base substitutions. What they did have, however, were mutations that would make all of these tumors RAS-like. That is, they are signaling through RAF dimers. They had class 2 BRAF mutations A common one is the K61E mutation, but there are others as well, codon 466. These actually signal via, they are RAS dependent, so they are by themselves not constitutively active forms of BRAF, but they require RAS to be superactivated. And you can see that we had patients with NRAS, KRAS mutations, and then fusions that also signal through RAF dimerization. Although the subset is low, the frequency of TERT promoter mutations did not differ between both groups. By contrast, the non-responders were enriched for BRAF V600E mutations, and they also harbored a number of additional defects, including mutations of mTOR pathway, mutations of epigenetic genes, primarily truncation mutations of SWI/SNF complexes, and I'll get to that at the very end of my talk, splicing defects, particularly RBM10, and those were the key differences. But above everything, what struck us was the copy number analysis, which is shown here in panel B. So the exceptional responders are shown in red, and you see there was one particular patient who had a significant broad-based, haplotype, haplotype picture with LOH at several chromosomal sites, but otherwise very few copy number changes. By contrast, the non-responders were markedly enriched for 1q, chromosome 1q amplification. And I will get that to that in detail in a second. This is, in terms of the statistical differences, is illustrated here. Here are your exceptional responders in panel C versus non-responders. And you can see.
给他这个关掉啊，就在这里，在这里等一下。怎么了？
I'm sorry, probably there's somebody probably needs to mute.
Yep. We apologize. Yep. Please proceed. Sorry.
Yeah. And as you can see here, the non-responders were significantly enriched for 1q amplification. There was no difference in the 22q loss, which was present in an equal proportion of exceptional responders and non-responders. So when we then looked at the RNA sequencing data from these patients, and we look at the principal component analysis, the exceptional responders tend to cluster more tightly together, whereas the non-responders actually are quite diverse transcriptionally. The actual histology did not actually segregate specifically with the PCA. It was primarily either their response to radioactive iodine, which determined probably the differentiation state that tended to determine their overall distribution here. So if we look at the score that has been derived by the TCGA for thyroid differentiation, which is an integrated score of 16 genes that are involved in thyroid hormone biosynthesis, the exceptional responders had a higher TDS than the non-responders, as you might predict. Also, if you look at the BRAF-RAS score, remember we talked about the tumors that are either more BRAF-like and RAS-like. As illustrated by the oncoprint, the exceptional responders were more RAS-like, whereas the non-responders were more BRAF-like. And this association between BRAF-RAS score and the ETDS, which is a enhanced thyroid differentiation score that we developed in the lab computationally to enrich the ability of this to make this distinction, showed a very tight correlation between these two parameters. So when we then look at the TDS score now as it relates to the copy number changes, we found this very interesting. If you look within the BRAF mutant cohort and look at those that had 1q gain, the presence of 1q amplification decreased the thyroid differentiation score to a greater extent than what you would find in the BRAF as a whole, which is illustrated here. Whereas 22q and those that were quiet had no copy number changes, were no different. 22q did not affect the thyroid differentiation score, so it was only the 1q amplification that appeared to do this. If you now just look at both gene ontology as well look at hallmark signatures of the RNA-seq, I'm going to actually just go through this very briefly. This is a busy slide. Here what you're looking at is the comparison in the TCGA on the top, in panel D here, between the BRAF mutant cancers that were quiet versus those that had 1Q gain. And as you can see, those that had 1Q gain had activation, the BRAF quiet compared to the 1Q gain. So if you think of it this way, 1Q gain would have a decreased expression of genes involved in cell adhesion and cell junction assembly. And this is consistent with prior data in the literature that had shown that 1Q amplification was associated with worse prognosis in thyroid cancer and presumably a more metastatic phenotype. But also notice that there is also a program of adenylate cyclase modulating GPCR signaling pathway, indicating that there may be some negative pressure by 1Q amplification on the key signaling pathway involved in regulating thyroid differentiation. In addition to that, if you now look at the comparison of the BRAF V600E quiet versus the 1Q gain, again, you can see that there is a downregulation of programs of MAP kinase signaling and of epigenetic regulation. So it seems like this program, which is the polyclonal repressor complex, and this one, which is the SWI/SNF complex, seems to be somehow engaged by chromosome 1Q gain in such a way that you would expect to see a more compact chromatin state and less accessibility to thyroid transcription factors. So summarizing this part of my talk, metastatic thyroid cancers that show major structural responses to reactive iodine are enriched for mutations that constitutively activate RAF dimers, that is, RAS class 2B RAF mutations and RTK fusions, and are usually diploid. RA refractory metastatic thyroid cancers, by contrast, are enriched for BRAF V600E, have frequent chromosome 1Q amplification, and harbor lesions of genes in the PI3 kinase pathway, splicing regulation, and chromatin remodeling pathways. So let me turn now to a key transitional event in tumor progression in thyroid cancer, one that I know Ming Zhao has had a lot of interest in, and has contributed really important information on our understanding of the TERT promoter mutations in this disease. So the overall frequency of TERT promoter mutations in the thyroid cancer based on the TCGA was 9%. If you look at patients with more advanced thyroid cancers, clearly the frequency is greater, but this is the data from the TCGA repository, which primarily looked at tumors that were relatively low risk. But now if you look at poorly differentiated thyroid cancers, the frequency is 40%, and in anaplastic thyroid cancers, it's 73%. So clearly, TERT promoter mutations are enriched as we progress along the overall spectrum of severity of this disease. What we also know, actually from the original publication in melanoma, that showed that activation of MAP kinase signaling, primarily through induction of ETS proteins, appears to be able to specifically bind to the base substitution in the promoter that generates de novo ETS binding sites. Ming Zhao has done a lot of work in characterizing the specific ETS family proteins that are involved in this interaction. I won't get into that in further detail, other than to point that this association between BRAF and TERT promoter mutations is likely at least explainable in part because of this fact that MAP kinase activates the ETS family of proteins, and these in turn have the ability to bind to these mutated promoters. Consistent with what I just showed you, what we noticed when we looked at more advanced disease patients was that mutations of the TERT promoter were subclonal in papillary thyroid cancer, consistent that they were in an early stage of evolution, and they become clonal in advanced disease. Now, a recent study adds an important element to this information, which is this study from the CNIO in Madrid by Cristina Montero Conde, who actually was a former postdoc in the lab. And what she found was that when she looked at other components of the telomerase complex, including TERK in particular, as well as these others that are illustrated here, their expression is increased in patients who have progressive forms of differentiated thyroid cancer, as opposed to those that are, as compared to those that are disease-free. And she then came up with various signatures to try and demonstrate the prognostic potential of this, and in particular finds that the expression of TERK as well as TERT, TERK is actually the RNA template for telomerase, essentially provides additional prognostic information. But the reason I found this interesting is that there are many studies that argue that the expression of telomerase does not necessarily indicate that if it's increased that it will have a role in immortalization, but there are non-canonical effects of telomerase on a number of other properties, including DNA damage repair and so on. But the fact that other components of the telomerase complex are also simultaneously, their expression is increased, argues quite strongly, in my view, that immortalization is the key property by which TERT promoter mutations promote the pathogenesis of this disease. This is data from Ming Zhao showing also the very impressive prognostic impact of TERT promoter mutations in this disease. Here you're looking at recurrence-free survival in patients with BRAF mutations alone, which is shown in blue, versus those that also have a TERT promoter mutation, which is shown here in brown. So I'll stop there concerning papillary thyroid cancer, and let's turn to what we know of a rare type of thyroid cancer, which I'm only illustrating in this slide, which is the Hürthle cell carcinoma. So this is a disease that has puzzled the field for many, many years. These types of cancers are actually characterized by the fact that they are loaded with mitochondria. There's a marked sort of increase in the number of mitochondria. So these tumors are very oxyphilic in their appearance, very similar to chromophobe tumors of the kidney, etc. So there are other sort of types of cancers that also are characterized by this, abundance of mitochondria. There have been two studies, both published back-to-back in Cancer Cell, that have characterized the genomic features of this disease. And one of the key sort of findings was that there is a near total haploidization of the nuclear genome. That is, that all of these chromosomes that are labeled here in blue are essentially present as a single copy in this disease. So essentially, they've lost one whole complement of virtually the entire genome, with the notable exception of these three chromosomes, 5, 7, and 12, shown in black, that are always retain two copies, both the paternal and the maternal. We don't understand why, but this is a very intriguing finding. What sort of added fitness to the tumor is given by the lack of two chromosomes in each of these other sort of sites listed in blue? We don't know. I mean, one thought is it could actually augment the tumor suppressor sort of dysfunction if you have a gene such as NF1 or TP53, illustrated here in chromosome 17, that is mutated. It would be immediately homozygous. Having said that, the mutations that were found, by this I mean point mutations or small indels, that were found in known oncogenes, are illustrated here in this gray color, and they were relatively rare. So most of these abnormalities are primarily haploidization of the genome with very few additional mutations. Now, where we did find additional mutations were in genes that encode mitochondrial proteins, and particularly those that are in complex 1 in the electron transport chain. So dysfunction of complex 1 of mitochondrial function is quite a profound disruption in respiration. And this seems to be a common property of Hürthle cell cancers, such that these mutations tend to be homoplasmic, meaning that virtually all mitochondria in these tumors harbor these mutations. So mitochondrial mutations in cancer are not that infrequent, but because as every cell has numerous mitochondria, usually they are present just in one or two of the mitochondria, but not in the remainder, and does not affect overall respiration. This is different. Here you have all mitochondria, or the majority of mitochondria, harboring these loss of function mutations. So this combination, haploidization of the genome and mitochondrial dysfunction of complex 1, are the key disruptions that likely account for Hürthle cell cancer. A lot more that needs to be learned here, but there are a number of groups now doing functional studies to try and understand this disease better. It's been hampered by the lack of appropriate cell lines. So now there's only 2 or 3 cell lines from Hürthle cell cancer, and so maybe some progress will be made. So let me turn to genomics of the more advanced forms of thyroid cancer. And I have to apologize that I will primarily be presenting work from our group. There have been a number of studies, all of them relatively small. We have not had a TCGA-like effort for anaplastic and polydifferentiated thyroid cancers. So we rely a lot of information on relatively smaller series. So because many of them have been quite consistent in their findings, I'll show our data as well as the data from Brian Haugen's group. But of course, there are others that just, in the interest of time, I don't have, the ability to summarize. So this is a study that we used where we used the MSK Impact platform. This is a 341 cancer gene exome platform that is actually quite informative. So much so that, for instance, when we did our exceptional responder study that I just described earlier, we did both whole exome sequencing as well as MSK Impact, and we actually did not find additional lesions by whole exome sequencing that were relevant as compared to what we normally see with this platform. So overall, what we found was that altogether we had, I can't remember the exact number, but I think something in the order of 80 polydifferentiated thyroid cancers, about 30 anaplastic thyroid cancers. And again, you can see very similar pattern that what we showed with papillary thyroid cancers. BRAF mutations are very common, 45% in this series of anaplastic thyroid cancers, followed by NRAS mutations, some fusions as shown here, as well as a smattering of other mutations. Okay. Note the TERT promoter mutation frequency. It's 40% here. It was 9% in papillary thyroid cancer. And then a number of additional lesions I'll get into in a second. In anaplastic thyroid cancer, very similar. So, sorry, I misstated this. BRAF in polydifferentiated is 33%. In anaplastic, it's 45%. Here, the TERT promoter mutations are even greater, but the hallmark of anaplastic thyroid cancer is P53 mutations. These tend to be sort of very dramatically enriched. They're only 8% in polydifferentiated thyroid cancer, and we don't see them in papillary thyroid cancer, or very rarely. Let me break this down. Here, what you're looking at is the tumor mutation burden. Of course, we're only looking at 341 genes, but think of this as being relatively proportional to what you would find by whole exome sequencing. So as you see, the polydifferentiated thyroid cancer are not particularly TMB high. They're TMB low to intermediate, and a little bit higher in anaplastic thyroid cancer, but still much lower than tobacco-related diseases or UV-related diseases such as melanoma. Having said that, if you look within polydifferentiated thyroid cancers at the impact of tumor mutation burden, those patients that had a tumor mutation burden that was above the median were usually older patients, they had larger tumor sizes, and they had more frequency of distant metastatic disease. Here are the various mutations in the MAP kinase pathway, mostly the BRAF and RAS mutations. Now, when we did this study at Memorial Sloan Kettering, we had a particular definition of polydifferentiated thyroid cancer that also encompassed patients who looked like had tumors that looked like papillary thyroid cancer, but happened to have a very high mitotic rate, and as well as necrosis. So this was irrespective of their growth pattern. Now, the WHO disagrees with this, and they basically call these high-grade papillary thyroid cancers. So I mentioned this because, as I'll show in a second, probably if we were to do this today, we would not call these tumors polydifferentiated. The definition that is accepted for poorly differentiated is called the Turin proposal, and that's a particular growth pattern, a solid growth with high-grade features. Now, these are the Memorial Sloan Kettering definition. These are the high-grade papillaries. Not surprisingly, 81% of these had BRAF mutations. So the reason we call them poorly differentiated is because their overall prognosis is identical to that of the Turin proposal cases. So their disease-specific survival and their progression-specific survival overlaps with these. So even the biological behavior is similar. However, histologically, the pathologists would like to keep on calling these papillary thyroid cancers, but they call them high-grade. Now, what is important is that BRAF and RAS dictate distinct tropisms for metastasis. Now, we've known this, I'm sure Ming Zhao has known this too for a long time, but this, I think, illustrates it in a very convincing way. That BRAF mutant tumors, as compared to those that are BRAF wild type, have greater frequency of nodal disease. That's N1A and N1B. But they do not have a higher frequency of distant metastatic disease. By contrast, those patients who have RAS mutations very, very rarely have nodal metastasis. In fact, this might even be an overrepresentation, but they frequently metastasize at a distance. So when NRAS tumors are bad, they actually metastasize to lung and bone. Now, in addition to these lesions, you can see that there is a stepwise increase in mutations that affect these pathways, the PI3 kinase pathway, the switch SNF chromatin remodeling complex, histone methyl transferases, as well as mismatch repair deficiency genes. And those that are significantly greater in anaplastic thyroid cancer are illustrated here. There is also mutations, as here illustrating primarily other differences between the groups. NF1 mutations in our series were more common in anaplastic. So were PIK3CA and PTEN. These are PI3 kinase pathway mutations, whereas fusions are more common in poorly differentiated. And even though they exist, not in our series, but other groups have reported them, in anaplastic thyroid cancer, they are less frequent. Now, this is the study from Nikita Pozdayev and Brian Haugen at University of Colorado. They used a platform that is called Foundation Medicine. The difference in this also is a cancer exome platform, but they do not subtract germline mutations. So MSK Impact actually samples normal tissue, usually blood, and then the tumor in order to subtract germline abnormalities to only report somatic mutations. Foundation Medicine cannot do that. So if there are mutations that are inherited in the germline, they will appear as cancer mutations. Having said that, they combined both our data, so they integrated our data, as well as the data from Foundation Medicine to create this hierarchical clustering where they broke down anaplastic thyroid cancers into 5 clusters. And these are either BRAF mutations associated with PI3K activation or SWI/SNF activation in cluster 1, those that have loss of function of CDKN2A or CDKN2B, P16 mutations, so these are clearly cell cycle deficient tumors. And the third is there are 2 clusters driven mostly by RAS. The second of these is also associated with certain mutations in receptor tyrosine kinases, which we did not find. And the final is one in which they have a scarcity of MAP kinase or PI3 kinase drivers other than PTEN, but then are driven by NF1 and RB1. So this is like a tumor suppressor subset, and that has a high tumor mutation burden as well. So returning to our study, this is the TERT promoter mutation difference, and illustrated the P53 defects I mentioned. So now let me just turn very briefly to a study that actually was, this is from Inigo Landa's study on PDTCs and ATCs. And just to illustrate that we found a relatively high frequency of mutations of EIF1AX. This is a component of the translational pre-initiation complex, and I will describe this in a little bit more detail in a cartoon in a second. So what we found was that these mutations were markedly enriched in advanced disease. So in papillary thyroid cancer, the frequency of these mutations is 1%, and they were actually not associated with any mutations in the MAP kinase pathway. By contrast, in anaplastic and polydifferentiated thyroid cancers, the frequency was 10%, so it's enriched tenfold. And they are universally associated with RAS mutations, almost all of them, I think all but one. This is also true in the cell lines, as well in a prior study from a whole exome sequencing study of anaplastic thyroid cancers. So in advanced thyroid cancers, EIF1AX mutations and RAS are very strongly associated, as illustrated here. The presence of EIF1AX mutations is associated with worse outcomes as compared to wild-type tumors. So this is a study that Jnana Krishnamurthy published in Cancer Discovery a couple of years ago. It is a relatively complex mechanism, but let me see if I can very briefly summarize what we understand from this. So, and I bring this up as well because as Dr. Gore's group does proteomics, the 3 of the cell lines that you will study actually harbor combined RAS mutations and EIF1AX mutations. What Jnana found was that the EIF1AX, which is a component of the translation pre-initiation complex, actually increases the overall affinity for components of this complex. So think of it as a complex that is harder to dissociate. When it does this, the role of this complex is to actually bind to the messenger RNA and essentially identify the initiation codon, the AUG, the methionine codon, which will actually drive translation. When this mutation is present, there's a tendency for this identification of the initiation codon to be less efficient. Now, and so overall, initially, the impact on translation, you would expect it to be dampened. However, paradoxically, there is one particular transcript, the ATF4, which actually has 2 open reading frames, one of which is a dysfunctional one, so and which is the one that normally operates with an intact pre-initiation complex. And that just leads to a truncated peptide that does not really sort of drive expression of the entire protein. But in the context of EIF1AX, the second initiation site is actually recognized, and it drives increased translation of ATF4. This, in turn, through a mechanism proved by other groups, induces a number of different things. First of all, it activates a stress response on the gene expression program that I'll describe in a second, but it also increases the expression of this particular phosphatase, GAD34. And this phosphatase dephosphorylates EIF1A2A, and this happens to be a major gatekeeper to induce overall global protein synthesis. So when EIF1A2A is actually dephosphorylated, global protein synthesis ensues. Now, in addition to this, ATF4, as well as MYC, and I will get that into a second, induces the expression of amino acid transporters, which are illustrated here, LAT1 and ASCT2. These essentially transport essential amino acids and glutamine, which are then able to directly activate the mTOR pathway, mTOR1 specifically, which then feeds further to induce global protein synthesis. So in addition, through a mechanism that we demonstrated, but we could not understand precisely the underpinnings of at this point, EIF1X increases the overall stability, of c-MYC. And because mutant RAS also increases MYC stability, this adds to this strong induction of amino acid transport, all of which feeds into this global protein synthesis increase, which we believe is a central step that allows this cooperativity between these 2 oncogenes. So let me just turn to another set of defects that we find in patients with advanced thyroid cancer. And these are mutations in components of the switch-SNF chromatin remodeling complex. listed here in these various columns here and rows are the various mutations that are found in thyroid cancer of subunits of the SWI/SNF complex, R1A, R2, R1B, etc. Most of these, as you can see in black, are truncation mutations. So clearly these are loss of function, and this is illustrated here also in the lollipop plants, which, consistent with what we think as tumor suppressor genes, you can see that they occur randomly almost across the entire sequence. So what caught our attention here was about the importance of this was driven in part by a prior study that we had done in the lab when we were looking for functional evidence of genes that cooperate with MAP kinase drivers to induce thyroid cancers. And for this, we were looking for drivers that cooperate with oncogenic HRAS. So we had generated a HRAS knock-in model, and we knew that that was insufficient to induce thyroid tumorigenesis. So we thought we could use this as a way of using an unbiased forward genetic screen using the Sleeping Beauty transposon mutagenesis system. Let me explain what this is. So this system is essentially constituted of a mouse that contains a concatemer with 358 copies of a transposon element, which is illustrated here. This transposon element can integrate randomly in the genome at AT or TA dinucleotide sites. So if you then have a transposase, which is an enzyme that is able to liberate these transposons, it can then integrate into mouse DNA. And when it integrates into the DNA, it can either activate an oncogene because it has promoter LTR elements that drive transcription, or it can interrupt the sort of sequence of a tumor suppressor gene. So it represents an unbiased way of trying to identify genes that cooperate with other drivers to induce tumorigenesis. So this is the example. This is a mouse where you have the HRAS G12V allele. In this case, we drove it to homozygosity. So think of this as 2 copies of a mutant HRAS gene expressed under the endogenous gene promoter. And this is insufficient to induce thyroid cancer. But when we actually now cross these mice with mice that contain the transposon concatemers, as well as express the transposase in a Cre-inducible way, so it would only be expressed in the thyroid, then we actually had a number of mice that developed, tumors, either these relatively small nodules, as shown here, or bona fide polydifferentiated thyroid cancers illustrated in the other picture. And this is the table that summarizes the data. And you can see here, this is the relevant piece here. If we just look at the HRAS homozygous animals, out of 90, only 2 developed nodules by H&E. So the overall frequency was 2.2%. But in the presence of the transposons, it increased 7-fold. So then what we did is we sequenced the genome of these mice, of the tumors of these mice, to find out where had these transposons integrated. And as a sanity check, we know from data from Antonio De Cristofano's group that if you combine RAS with PTEN mutations, loss of function mutations, you get thyroid cancer. And indeed, what we found was that there were numerous transposons in many of these tumors that actually had induced tumorigenesis through loss of function of PTEN. But then there were a number of others, among which were mutations in SWI/SNF complex genes. And in fact, if you do an overall analysis of what these transposon integrations had in common, mutations of chromatin modifiers were at the top of the list. You can see here DNMT3A, this is involved, of course, in DNA methylation, MNL5, this is a lysine methyltransferase, ARID1B, ARID2 components of SWI/SNF, and so on. So with this in mind, our interest in SWI/SNF mutations was definitely peaked when we saw that they were quite prevalent in advanced disease. Not only that, they were subclonal in papillary thyroid cancer and became clonal in advanced disease. So this again pointed to the fact that they were probably important in tumor microevolution. And We know from other cancers that mutations of components of this complex in certain diseases, like for instance in ovarian and endometrial carcinomas, bladder cancers, and so on, tend to cluster around individual components. So ARID1A mutations are common in these particular cancers, whereas for instance in rhabdoid cancers, it's only SMARCB1, in renal cell cancers, it's PBRM1, and so on. In anaplastic thyroid cancer, they're distributed among 4 or 5 different genes. And this is also the case in the cell cycle encyclopedia. So in order to study this, we generated mouse models, and don't fear, I will not go into detail into this study that was published last year. I just want to give you the broad outlines of this. So we have used this knock-in mouse of BRAF V600E, which is Cre-inducible through thyroperoxidase Cre. In addition to this, we also have a YFP allele, which allows us to track the tumor cells in the mouse. And then we cross these to homozygous loss of ARID1A, which would disrupt the complex called BAF, ARID1-2, which would disrupt the complex called PBAF, and SMARCB1, which disrupts them both. And what we find is that if you have a BRAF mutation with either ARID1A mutations or SMARCB1, these animals die sooner. And if you look at their overall histology, whereas the BRAF mutant cancers give papillary thyroid cancer in the context of ARID1A, ARID2, or SMARCB1, they develop a more advanced disease. Surprisingly, the ARID2 mutants, even though they are quite mesenchymal, their overall survival was not significantly different than the BRAF itself. And you can see this in the tumor volume. The BRAF mutants, of course, are quite large tumors, but they're even larger if they have ARID1A or SMARCB1 mutations, but not ARID2. And I'm going to summarize the conclusions of this study in this cartoon here. So if you have a thyrocyte and it has develops a BRAF mutant tumor, these become metastatic. Normally what we find is that the chromatin is compact and essentially inaccessible to thyroid lineage transcription factors. And because of that, you cannot get transcription of the sodium iodide symporter, thyroglobulin, thyroperoxidase, and so on. So patients with BRAF tumors tend to be radioactive iodine refractory. Through the studies using RAF or MEK inhibitors that I described earlier in my talk, this chromatin actually becomes accessible to the lineage transcription factors, and they can now drive the thyroid differentiation program, and in a subset of patients, we get restoration of radioactive iodine avidity. If you, by contrast, have mutations of key SWI/SNF components, then although the chromatin is compact to start with, as I showed before, even in the presence of RAF or MEK inhibitors, you cannot actually get a restoration of open chromatin, because in the absence of SWI/SNF, you have occupancy of the histone marks by the polyclonal repressor complex, which results in histone lysine 3 lysine 27 trimethylation, which basically renders this chromatin inaccessible, and therefore there is no thyroid-specific gene transcription, and these tumors remain radioactive iodine refractory. So, in a nutshell, this is at least a simplified view of the key events associated with thyroid cancer disease progression. In papillary thyroid cancers, you primarily have mutations of the key effectors in the MAP kinase pathway, very few additional defects, TERT promoter mutations in less than 10% of tumors, IFAX mutations in 1%, and they are not associated with any of the MAP kinase mutants, and very little in the name in terms of copy number abnormalities. I am leaving out some other interesting findings, but this is, in a nutshell, what we think, the way we think of this disease. In poorly differentiated thyroid cancers, there's an enrichment for PI3K pathway mutations, as well as IFAX mutations, which now are associated with RAS mutations and drive a worse phenotype. And in addition, the TERT promoter mutations become more frequent. And we believe that because of its immortalizing function, is one of the factors that allows for additional accumulation of genetic defects. In anaplastic thyroid cancers, now you essentially see a marked acceleration of these events. P53 mutations happen. This is critical and a key transitional step to anaplastic thyroid cancer. Mismatch repair deficiency genes also become apparent, although in a small subset. And as I mentioned, SWI/SNF complex genes, also histone lysine methyltransferase genes are also mutated quite commonly. We're very interested in that. PI3 kinase pathway mutations. So these are the key events associated with the more advanced explosive form of the disease. So let me just close with summarizing very briefly some general thoughts on this other important oncogene, RBM10. Jnana Krishnamurthy, again, has been leading this work, and I want to bring it up in a slight unbiased way for Tian Nguo's group, because you will be having, I believe, at least 2 or 3 cell lines that have RBM10 loss of function mutations. And we are very interested, as you know, we'll be giving you isogenic lines to explore what proteomic changes you might find. So I wanted to give you a brief introduction about this. What we know about RBM10 is that it's a component of a key splicing complex. Primarily, it's involved in cassette exon exclusion events, such that in the absence of RBM10, you actually get exon inclusions. You get genes that would normally be spliced, exons that would normally be spliced out, that are now actually incorporated into the final transcript. There can also be other events, such as alternative 5' splice sites might be utilized. Most of these would be maybe in frame, but some actually may be out of frame and may actually encode proteins that could become neoantigens. The same is the case for intron retentions. That's also something that can happen, although less frequently. Now, we've been interested in looking at this in the context of oncogenic activation of the MAP kinase pathway for obvious reasons. And what we found is that it's associated with thyroid cancer progression and specifically metastasis. So is this common? The answer is no. I mean, it's a rare event. So across all cancers, 4%. Mostly, as you see here, loss of function, truncation mutations. If you look at the overall number in the MSK impact series, you have either deep deletions or truncation mutations, and they're associated both with BRAF and RAS mutations. So we see them in the context as well as with RET fusions. Now, the other reason we became interested in this was that Ian Ganley did this study published a few years ago, where he looked at patients who had died of thyroid cancer, but they did not die of anaplastic thyroid cancer. So he was interested in patients who had actually died of thyroid cancers that were either differentiated or poorly differentiated, but not anaplastic. And here, the incidence increased from 0.5% RBM10 mutations, that's 2 cases in the TCGA series, to 11%. So that's quite a striking increase. And here you can see the overall survival of patients with RBM10 mutations for those with a wild type. You can see it's significantly different. And even more impactful, at least in our thinking, even though this data came when we were already heavily into this work, this is a study published earlier this year by Nikki Schultz's group at Memorial Sloan Kettering that leveraged the 25,000 cases from the MSK impact series. And specifically, they call this the MSK MET cohort. They looked at events that were associated with metastasis, genomic events, across in a pan-cancer way, meaning across all tumor types. And when you look at thyroid papillary carcinoma, the only events associated with metastatic disease, CDKN2A, TERT, and I think this is primarily local regional metastasis, and RBM10. And in this case, it's distant metastasis. So we made this mouse model in the context of HRAS. We tend to use this one because it's a background associated with very low transforming potency by itself in the context of RBM10 loss of function. This mouse is one that we created in collaboration with Omar Abdul Wahab here at Memorial. And these take quite a long while to become tumors, but they do. And when they do, they develop metastatic disease, illustrated here, metastasis to the lung. And you can see the overall size of these tumors. So they're larger. They're not spectacularly larger, as you see in BRAF context, but they clearly are growing. This is in the HRAS heterozygous and homozygous context. And you see, quite significantly larger tumors than you see in HRAS alone. These are the 5 cell lines that we used in our isogenic system. And in this case, the parental cell lines from these 3 lines actually express RBM10 normally, have no mutations. And these we did knockdowns with 2 different hairpins. And these 2 cell lines, KTC1 and PE121410, have a autochthonous RBM10 mutation, okay, which results in loss of function, and which we rescued by doxycycline-inducible expression of RBM10. And we then did RNA sequencing across all of these lines, and then these were analyzed by Rob Gradley, who's an expert in alternative splicing at the University of Washington in Seattle. And you can see the primary events were primarily, as I mentioned, exon inclusion events, which is shown here in green, with the other types of defects being in a minority, but very consistent defects or types of defects in all 5 cell lines. So we also, as I mentioned, when we looked at the mouse model, we were also able to make cell lines that I'll show you in a second. But this is using here the parental cell line that has an RBM10 mutation, in which we have a doxycycline-inducible expression of RBM10. You can see even in the absence of doxycycline, it's slightly leaky. So there is some modest expression of RBM10, even in the absence of doxycycline when it's transduced. And what Niena did was to look at the overall mean velocity of these tumors in vivo. So we're actually doing a time-lapse imaging and looking at how these cells move over time. And as you can see, expression of RBM10, in this case in a dose-dependent manner, is associated with a decrease in overall mean velocity. And the converse is true if you, in a cell line that expresses wild-type RBM10, when you knock it down, you actually induce increased cell velocity. And that's illustrated further on the right panel, where you can actually see the trajectory of these cells. So in the parental, you can see the cells actually are moving in many different directions, but in the presence of doxycycline, they are essentially restricted from movement. And the opposite is true in the cell line where we knock down RBM10. As I mentioned, Niena developed cell lines from the mouse metastatic lesions that have RBM10 loss, and then induced RBM10 expression to rescue the expression in these, and then implanted these after transducing them with luciferase into the tail vein of mice. And those that actually lack RBM10 consistently developed lung metastases, whereas those in which RBM10 expression was restored had a much decreased metastatic propensity, as quantified here. So when we actually did the RNA sequencing in the mouse model, what we found was a association of a number of genes that were involved in extracellular matrix and cytoskeletal remodeling, many of which we have now characterized functionally, in terms of which particular exon inclusion events might be driving this. And I've left that information out in part because I would like our proteomics colleagues to, when they analyze these cell lines, to have an unbiased look at what sort of exon inclusion events they find. But we will obviously be sharing this as we continue to discuss our collaboration. One final slide, and I apologize if I went a little bit long. Something to bear in mind as one is doing proteomic analysis of cell lines is that, of course, in the tumors, these cells don't live alone. They're actually in the context of a very complex tumor microenvironment. And in the context of thyroid cancer, that is particularly complex, certainly in the setting of more advanced disease in anaplastic thyroid cancer. So this is work that we did in our lab, as well as with Jenna French at the University of Colorado, is a multispectral immunofluorescence of human tissue microarrays of papillary thyroid cancer, polydifferentiated thyroid cancer, or anaplastic thyroid cancer, where we use markers for myeloid-derived suppressor cells, M2-like tumor-associated macrophages, M1-like macrophages, T cells, etc. In papillary thyroid cancer, these are primarily when we say cold tumors, immunologically speaking, primarily enriched with T cells with a relatively low myeloid component. The polar opposite of this is anaplastic thyroid cancers, which we know from work from a number of years ago from our group and others, that they are very heavily infiltrated with myeloid cells, as shown here. So one question which is really important is what specific signaling programs arise from the thyroid cancer cells, and in the cell lines, many of them derive from anaplastic thyroid cancer, which drive this particularly complex tumor immune microenvironment. The other thing that I would mention is that when we actually did RNA sequencing and immune deconvolution of human papillary cancer, polydifferentiated and anaplastic thyroid cancer, and compared that to mouse models that we have created of these three histologies, what we find is that, first of all, encouraging to us as people who work a lot with mouse models, is that there's a very nice concordance between the overall immune context of human tumors and mouse tumors that are recreated based on the genetic abnormality we find in this disease. Note in particular the very heavily myeloid infiltrate that you see in anaplastic thyroid cancers, as we've shown as well by immunofluorescence. So I'm going to stop here, and again, grateful for your attention. I've mentioned the people in the lab who actually were involved in doing the experiments that I described, or a lot of this was a very broad overview. And I thank you very much for your attention, and happy to answer questions or discuss this further.
Thanks so much, Jim. Very, very nice and very systematic presentation of the thyroid cancer carcinogenesis. So any questions from the panel? Let me start with one very naive question. So compared to many other malignant tumors, thyroid cancer is relatively benign, except ATC. So from the oncogenic signaling pathways, do you see any major difference between PTC and ATC, or PTC versus other cancers, like melanoma? They also have BRAF, but they are so malignant.
Well, I mean, so the answer is yes, you're totally right. I mean, papillary thyroid cancers are usually quite indolent tumors, so much so that, as you may know, there is a current trend to actually not operate on small papillary thyroid cancer. So many patients who have papillary thyroid cancers less than 1 centimeter, or even less than 1.5 centimeters now, are being, in several groups, including our own, offered the option of not having surgery at all and just actually getting monitored over time. And so the basis for this, I mean, we really don't know. I mean, what is it that makes BRAF mutant or RAS mutant tumors that do not harbor TERT promoter mutations? I think it really is important to mention that, tend to be indolent. Could it be oncogene-induced senescence? So one possibility is that when you activate the MAP kinase pathway, you're actually inducing a senescence program. There's evidence from that, from studies from Yuri Bharti and a number of other people, that when you activate MAP kinase signaling beyond a certain threshold, that you actually get DNA replication fork disruptions that essentially arrest the cells, and they're unable to progress through the cell cycle, and they therefore become senescent. The other alternative is that there's a host effect. So is it possible that there is an immune recognition of the foreignness of the BRAF mutant thyroid cancer, and that there's an immune response to this that essentially prevents this from growing any further? Once you get additional defects, TERT promoter defects, CDKN2A mutation, I mean, other events that cooperate with BRAF, then it's no longer a disease. And regarding your comparison with melanoma, remember, most benign nevi actually have BRAF mutations, right? So you do have sort of a very similar paradigm there, where if you just harbor the actual mutation in the pathway by itself in an isolated way, it's insufficient to drive cancer.
Yeah, probably I can conclude that looking at only the mutations cannot explain the story. The seeds is the mutation, but you also look at the soil. maybe the proteome have an impact. Not only the proteome of the tumor cells, but also from the microenvironment.
Yeah. I see that Goma has a question.
Hi. Sorry, I'm still in my scrubs because just finished in the OR actually. I had two questions actually to ask. One was something you alluded to recently about the fact that we are not operating on a lot of our patients with thyroid cancer, which I'm grateful for, and yet you mentioned that there were a proportion of them with subclonal mutations in both the SWI/SNF complex as well as TERT. Do you think that these should be the biomarkers we use to identify those that require earlier intervention? Are these the clones that are going to come back as your metastatic tumors? So that's my first question. My second question is something different about the RBM mutations and splice variations. I think it was Omar Wahab who recently published data showing that splice events can serve as tumor antigens. So have you looked at those RBM mutants and asked whether are you seeing more T-cell infiltration, more evidence of new antigen presentation in those? So those are my two questions.
Both excellent questions. So first, I totally agree with you in terms of I would like to see, I mean, in a perfect world, and if resources were infinite, I would sort of like to see that in active surveillance programs, that we actually do some more comprehensive, deeper sequencing, because I worry about the tumors that have subclonal mutations. We have no data, as you know, but in order to prospectively say, okay, these tumors are going to actually progress, because once you've sampled them, that's essentially the end of the story, right? So, I do worry about that. It's a very, very small proportion of the tumors that harbor these subclonal events. If you look at the total number that I found with SWI/SNF, it was like, I can't remember, but it's like 6, 7, 8 out of 500. So we're talking about 1% frequency, and for TERT it's higher. So for TERT promoter mutations, I think there would be a greater justification for considering excluding those from active surveillance programs. And the question is whether the sensitivity of conventional genotyping approaches is sufficient to pick up these subclonal events, right? So there I would argue that active surveillance could be improved by genotyping for that, yeah. Concerning your second question, we are very interested in exactly that point, and we are now looking, certainly in our mouse models, we just don't have enough clinical samples from our patient population that we have actually been able to get our hands on, at least so far, to look at the immune infiltrate. But we certainly can do this in a rigorous way in the mouse models, and we plan to do that. That's definitely sort of very much what we want to do. Yeah.
Thanks.
Well, may I also join, to ask question here. Jim, that's a wonderful talk. Very enjoyable, wonderful, very elegant talk, as always. I have a few questions back to the first part of your discussion today. Speaking of BRAF and RAS classes, right, the concept was coined a few years ago with that cell paper. Now, quite a few years have passed, right? So at that time, I think the phenomenon was very clear. The thought was straightforward, and maybe simple. So at that time, we thought our tumor could be classified in a molecular manner versus classical pathological manner, right? So, but today, even today, I think after all, we are looking at the phenotype, at the clinical presentation. That is, we are still looking at what histological, pathological type of tumor is. Specifically, we still look at whether the tumor is papillary thyroid cancer or follicular thyroid cancer or anaplastic thyroid cancer. There's still, how you use the type of classifications in your study and in your presentation today. So then the question is, how practical do you think it is to classify thyroid tumors into BRAF and RAS classes? In other words, how can we translate these two genetic-based molecular classifications into clinical practice? Now, before you answer me, let me add one more comment. I said it was simple at that time, right? So BRAF, RAS, you find it out. Oh gosh, very interesting, exciting. Tumor that had BRAF mutation, right? Had quite different clinical patterns or pathologic patterns, and even other molecular patterns compared with a tumor that had a RAS mutation. But today we know there are many other genetic alterations, not BRAF, not only RAS also, right? And in your study, you have so extensive study over the years on the genetic patterns and all kinds of thyroid cancers, right? So we know BRAF mutation tumors themselves are very different genetically. They probably uniformly have BRAF mutation, but they have very different other genetic patterns, right? We know most papillary thyroid cancer that have BRAF mutation are doing well clinically. Only a small group that are not doing well. That's because they have other genetic alteration, like terminal mutation, for example, right? And also now you discover EIF1AX, right? That's mostly associated with the RAS mutation. So for RAS mutation tumors are not uniformly the same. They're very different because they have other mutation, as I said, like your EIF1AX, right? And now you also have the RBM10 mutation. That's very interesting, by the way, very exciting discovery. Now that's also occurs in a small portion of thyroid cancer, but those patients with that type of cancer are not doing well. They may not, the coexistence, you said that coexistence also with second mutation. I don't remember. Does it coexist with RAS mutation?
They coexist with both RAS and BRAF, yeah.
Oh, right. So now that means a lot of RAS positive tumor do not have this RBM mutation. And also likewise, a lot of BRAF mutation positive tumor do not have this RBM mutation, right? They may behave differently. So 80 years ago, or maybe 80 years ago, when that Cell Power published that, I think at that time, the rationale was relatively simple. Today, given the knowledge that there are many other mutations that make a great difference, not just BRAF RAS. So then is it practical to classify tumor into BRAF and RAS classes? So that's that's my question here. I want to see how you think.
Thank you. I mean, well, basically, I agree with everything you said. I mean, we in our clinic, and even people who are primarily clinicians, like say Mike Tuttle and other people who have been very influential in the clinical management of the disease, now think in terms of, is this a BRAF-like or a RAS-like tumor? So if we have a patient who presents with follicular variant papillary thyroid cancer, we are more worried about whether or not the patient has vascular invasion or not in order to make determination of what we're going to do next, right? If we have a BRAF-like tumor, we make the assumption that we're going to have to be monitoring the neck very carefully, over time. But if they have a RAS-like tumor, we worry less about doing ultrasounds in the neck, and we're more worried about the possibility of distant metastatic disease. And you're right, not all RAS-like tumors are the same, not all BRAF-like tumors are the same. And that's why understanding of how these co-mutational events really contribute to disease, to the biology of the disease and to the clinical behavior. So I get frequently asked because we do MSK impact on a subset of these patients when they come in, what do I do when I have a BRAF plus this, or what do I do when I have a RAS plus the other, right? So, and we have answers for some, but not all. What do we do with a patient who has a BRAF and a CHEK2 mutation, for instance? The answer is we don't know. It hasn't been modeled. There's not enough of them for us to actually look carefully at patient series and so on. So, and you have done a wonderful work in terms of getting a number of institutions to collaborate with you as you do your epidemiological, studies in terms of follow-up. I think one good question that could be asked is, now that more cancer centers are doing complex panels, is to begin looking at prognosis with co-associated events, right? So if you do have one of these rarer mutations, maybe if you're now collaborating with 7, 8, 9, 10 institutions that have a decent number of patients studied with these panels, what is their natural history, right? So I think we need that data, because we don't have answers. We have answers for some, but not all.
Yeah.
But basically, otherwise, I agree with what you said.
Mm-hmm. Yeah.
Sorry, can I insert one question?
I agree completely with you, all you said, because the BRAF classification become, I think, very popular. Even in the WHO classification for thyroid tumor, the most recent edition published this year already formally use RAS and BRAF classifications.
Mm-hmm.
So I suspect, I actually doubt whether this classification is practical or not, right? So not surprising you told me that your clinical practice at your institution, actually people do not use this, right? But let me ask another very quick question. Your discovery of Q1 gain together with BRAF mutation, very interesting. That's now I will identify a special group, right, of patients who have lost reliability, right? So my quick question is that without Q1, with the Q1 gain, you probably have many mutations, many genes amplified there, right? So I wonder at this time, I hope you have noticed any single gene or something that is targetable. Hopefully that play a major role in the loss of reliability, right? So the future is not just targeting the MEK, BRAF to try to restore the reliability. So something perhaps more targetable, more specific, hopefully through your recent discovery.
Thank you. I mean, no single gene, Mingzhao, that we can find so far, but a genetic program, like I showed in the RNA-seq data, this but we're very interested in that, and we're trying to understand it better. But we've been through the list of genes and that are overexpressed as a function of the 1Q amplification. You can mine that from TCGA. You can find it yourself. I mean, there and we've been asked that question by the reviewers of the paper that we think is going to be coming out soon, and we don't have an immediate answer to it, but we're obviously thinking about it. But it's yeah. Thank you. Yeah.
Well, thank you. Congratulations. That's very exciting discovery in this thyroid cancer field. Well, thank you, Jim.
Thank you. Tianan, you were going to say something.
Yeah, I was sorry, Popcorn. I know you have raised your hands. I want to ask you. You always talk about the mutations. If have you done single cell sequencing for those tumor cells in a specimen? I guess you will see some heterogeneity. I don't know how much degree. And then when you talking about mutations, you're talking about the bulk tissue. Can this represent the behavior of the tumor over many years, because the tumor normally grows relatively slow? So what's your view on this?
Yeah, so we've done actively working on single cell sequencing experiments, mostly in mouse models. And of course, those are biased because we know what genes we introduce, right? So in terms of the impact on the genetics of the disease, it's not a particularly clarifying model, but in terms of drivers of tumor heterogeneity that are independent of the genetic drivers, how the tumor evolves once it initiates, even though all cells might harbor the particular driver, they then actually sort of go through different transcriptional states. And we are very interested in understanding those, both in their sort of, natural history, as well as when you put selective pressure on them. So when you inhibit a key driver pathway, how do these tumors respond? How does the tumor microenvironment respond to that? There have actually been several really good studies from China recently that have done single cell sequencing analysis of papillary thyroid cancers particularly, and looking at overall transcriptomic trajectories and so on. So we are doing something very similar, but in more controlled settings, or more in the mouse context, right? So, but in terms of looking at overall genetic trajectories, there's work from Tobias Carling in the United States, who's been interested in the papillary thyroid cancer to anaplastic transition and within individual specimens, so by sampling different sites, and has developed, overall, evolutionary trees to look for overall trajectory of mutations. Does an anaplastic start almost de novo, or does it actually go through a sequence of transitions before it actually evolves? So we have not done that type of work, but others have. Tobias Carling is one example.
Okay. Prof. Sorry, I have another related question. It's a brainy question. You're talking about the tumorigenesis. We know the disease incidence is so high. Is reported maybe probably 50% of the adults have thyroid tumors. But I think more people care about who will become really malignant, become metastasis. So then from the mutation, do you think the mutation that you're talking about, BRAF or those RAS, are the key for the tumors to become really malignant, or others like RBM10 and others, or some we don't know?
I think that if you just have a BRAF mutation, it is one of the sort of quandaries in this. Normally, we think, is one gene sufficient to induce tumorigenesis? In the context of RAS, the answer is clearly no. So if you actually sort of make mice with a RAS mutation expressed at endogenous levels, even through the lifespan of the mice, the majority of them will not develop thyroid cancer. Some might develop a little nodule, but it's very, very infrequent. In fact, in the context of BRAF, it's a little bit more controversial in that when you introduce a BRAF mutation, they always develop a papillary-like nodule, right? So a papillary phenotype is always there. Pathologists will always call that cancer. Now, would you think that a tumor that is a BRAF mutant tumor that remains indolent, does not change in size, sometimes over the lifetime of the patient? I mean, there's a paper that Dr. Tuttle just published looking at overall evolutionary trajectories of papillary thyroid cancers. You have to assume 60% of those are BRAF mutant in all likelihood. And over like 10, 15-year period, the same thing by the data in the Japanese data, there's even longer in terms of its overall follow-up. These tumors actually are not growing, right? So we call them cancers, but they're not behaving in terms of cancer metastasis and invasiveness like true cancers, right? So it suggests that it's BRAF plus some contextual change. Could it be a second mutation? Yes. Could it be a host change? Maybe. Could it be TSH levels that actually cooperate in some way to actually drive programs? our first study of a BRAF mutant mouse model, what we found is when we deleted the TSH signaling pathway, we actually attenuated the penetrance of the disease. So there is like a contextual piece here. So I think single mutations need something else.
Great. Even without the BRAF mutation, there are still some ATC, lot of ATC without BRAF.
Oh, well, there are ATCs without BRAF and without RAS, but I mean, they're rare. I mean, fusions would explain some of them, but some others are primarily a combination of tumor suppressor genes, P10, RB1 loss, P53. That's a very bad combination, regardless of whatever cell type you put in there. If you have those three genes deleted, there's not too many cells that will not sort of behave really badly.
Thank you. Prof. Kong, sorry for the delay.
Okay, thank you. I just have two short questions for Dr. Fagan. The first question has to do with the enhanced thyroid differentiation score that you mentioned briefly. It seems to me that that could function as a kind of functional readout of the tumor. And my question is, have you looked at the performance of that score with other aspects of tumor thyroid carcinoma phenotypes, either biological phenotypes or the clinical associations? Because it would seem to me that underlying all the complexity, what you really care about ultimately is what it's doing. And it seems to me that you do have a functional readout.
Right.
That's
very good.
My second question is related to that, and it's a short one. Knowing what is known now from your group and other groups of what is associated, the molecular abnormalities associated with the loss of avidity for radioiodine uptake, do you think there is a plausible pathway to try and redifferentiate thyroid cancer cells to take up radioiodine again?
Let me thank you. Both very good questions. So the enhanced thyroid differentiation score, what we did here is we looked at genes based on TCGA that were computationally strongly associated with the core 16 gene TDS, either because of being overexpressed or underexpressed. And so it added an additional number of genes to the list besides the 16. It performs slightly better than the core TDS. And part of it as well is that within that 16 gene TDS, there were certain genes that probably shouldn't be there. I was part of the TCGA group, and this was a group of experts that we basically said, okay, these are the 16 genes that would probably, we told the computational biologist, don't do a unbiased look at the gene expression profiling. I know we have some biology here that you could base this on. And among those, for instance, was TR-beta, the beta receptor, the thyroid hormone receptor. It was based in part of Shuyan Chen's data that showing that this TR-beta was an important thyroid cancer oncogene, but it turns out TR-beta is extremely low across the board and doesn't add much to the TDS itself. So knowing that, what we did various permutations, and we came up with this enhanced TDS, right? And it performs slightly better. But in answer to your question, we have used it in the context of the clinical trials that Dr. Alan Ho has been doing on redifferentiation, because we biopsy samples, patient samples before or after they're treated with the interventional drug, say a MEK inhibitor or a RAF inhibitor or a combination with a HER3 inhibitor or whatever. And it performs maybe slightly better in terms of distinguishing a differentiation state versus a non-differentiation, but it's not a huge difference. But then aside from that, we have not used it, say, to ask the question, is a RET mutant case compared to a BRAF mutant case more or less, etc. We have not done that type of work. But it's it would be reasonable to test. The second question is redifferentiation. Well, I mean, I am a little concerned about this, even though these studies were originated by our group. And there have now been, very good papers published by, the group of Sophie Lebouilleux in Paris, and a number of other groups, Wolfgang Weber in Germany, besides our own group, the group at MGH, validating the fact that, yes, you can restore radioactive iodine avidity in patients with BRAF or RAS mutant tumors, mostly RAS mutant tumors, no surprise, because you can ratchet up their iodine incorporation. In some cases, that you can then give radioactive iodine and shrink the tumors. But we don't have long-term data. So the only thing we have is the outcome of a short course of drug that we give to redifferentiate the tumors, a single dose of radioactive iodine, and what happens 6 months later. We need to know what happens 1 year, 2 years, 3 years later, right? Because one risk is we're treating the differentiated component of the tumor, but if there still is a subset, a subclonal group that is less well differentiated, how is that going to behave when we remove the tumors that can redifferentiate? Will it sort of take off, for instance, and lead to worse long-term outcomes? So the unfortunate thing is that this approach is now being used clinically all over the world. So I hear from colleagues when I give talks, in different parts of the world, that they did redifferentiation on this patient or that patient, because when they don't have anything else to offer their patients, they look at the literature and say, we should try this. I hear, mostly good news and people saying, okay, this worked and so on. But I don't like to recommend this, because we need the long-term clinical trials to demonstrate benefit with appropriate arms to show that this is beneficial.
Thank you. Thank you very much.
You're welcome.
Hey, Dr. Fagan, I have a lot of questions. I can pick the most two confused questions for me. The first is whether the TDS score and the BRAF-RAS score can guide the clinical decisions. And the second is, I think these two scores are related with the phenotype. And why, if I was a histopathologist, I would say I didn't need to calculate these scores, because I can read the differentiated score and the BRAF or RAS-like tumors by histopathology, by the HE staining. So why we need these two scores? And the third questions I have to ask is, these scores are based on the genes, and I would like to establish a new calculator based on proteins, and whether the proteins can be, I think the proteins are more related with the phenotype. And if these scores are really, really important and useful in clinic decision.
So let me ask you first, Dr. Sun, are you a pathologist? No. Okay, just out of curiosity. Okay, so the answer to this is, first of all, why are TDS and BRAF-RAS score not used in the clinic? The reason why is right now primarily a logistical one. I think we don't do RNA-seq or even targeted RNA sort of measurements on patient samples. So it's not been tested. So if you were asking, would it make sense to do a thorough differentiation score and a BRAF-RAS score in a patient sample, and then based on that determine whether or not they'll respond to radioactive iodine prospectively? Good luck in designing that study and getting it done, but it would be a very interesting study to do. In answer to your question about the pathologist, I think what you're saying is in a way, I think pathologists can give us a very good approximation of this, but I don't think they have the precision to be able to tell you this with certainty. And the third question is the answer to the world proteomics, which is, I think, what you're trying to say, and which would satisfy Dr. Guo very much. But the answer is proteomics is also another layer here, because it's not just is the protein made, but where is it, right? Is it at the, if in the case of the sodium iodide symporter, is it at the plasma membrane? Or what if it's mislocalized? So simply doing proteomics by itself will add enormously to this. We wouldn't be having this conversation and this meeting if I didn't believe that that's true. But it won't be the final answer. It'll be one more important layer in the answers we have to provide.
Yeah. By the way, we also have now spatial proteomics that we can analyze the proteome in different regions, different cells.
Mm-hmm. Yeah.
Yeah, thank you.
So, Niana, do you have anything to add? I know you are doing very exciting research on RBM10.
Yeah, no, to Jim, no, obviously. Yes, so I just would like to add on RBM10, maybe. Yes, we are on our active collaboration. I'm looking forward to analyze those differential splice events. So I realized that we can analyze at the isoform depth in the proteomics, right? So this kind of give us an advantage to know how differential isoforms could be detected that are expressed in the protein. We do have the data at the RNA level, but as Yating mentioned, I mean, we need to get some proteomics level to get it to relate that functionally. So that is where we would like to collaborate with you. And yes, we're happy to deal with that. Yeah, that's what I have. And obviously, no, nothing for Jim. Thank you for having us.
And then just a quick one on just to follow up on what Niana just said. Are you guys doing immunopeptidomics? Are you pulling down HLAs and looking at splice variants? Because that would be a very interesting thing to do in the PVRM, in the RBM10 mutants, right? With regards to my last question with to Dr. Fagan earlier, to pull down and see whether you're actually seeing these splice variants coming out on the surface and being presented by the HLAs.
We haven't gotten even close to that, Gopa, to be honest. I mean, I think the first thing we want to do is do we see any evidence that there is a different immune context in these tumors compared to the appropriate controls, right? So, and we actually we have focused almost entirely so far on cassette inclusion. So all of Niana's work has been characterizing a suite of proteins that have inclusion events that we thought were functionally consequential, and that he has demonstrated that that is the case. But we have not gone into our database and said, okay, now let's look at the proteins where there's a in-frame change based on the splicing change, and what new epitopes are they generating, and therefore how is that altering the TCR states and so on. So these would be all very, very good questions that we're only just beginning to touch on. Yeah.
Great. So Jim, actually now we're going to have a 2 hours talk and discussion. It's really a great webinar. And we have now doing live streaming, and in one of the platform we have over 3,700 views. And there are quite a few questions there, but I don't think we have time to elaborate that. I can email you the questions later, and probably we can then send your answers to those audience.
Okay, I mean, please don't email me 3,000 questions, but
yes, thank you so much for the thyroid. It's like a textbook to me, because I'm not in this field. I'm actually working on proteomics. But our interest is to use our technology to help you to understand thyroid cancers. I think from your presentation, I know a lot, but I have even more questions. There's so many things which we really do not know about thyroid cancer. There are so many clinical practices which are supported by some data, but probably since the data are not really that solid as we initially assumed. So they're like the host response to microenvironment and how the tumor evolves over time, and what's the real relationship with the mortality, with the survival. There's some very limited data. So I'm now working with many thyroid cancer researchers in China. Hopefully, through this discussion and through more interaction in the future, we can have better collaboration in this field and make the disease mechanism better understand in the future. Thanks so much. Thanks for all the panelists, and thanks you, Niana. And we initially planned to have you have another independent presentation, but
Sure. Yes.
let's discuss next time. Thanks so much.
Thank you.
Thank you.
Bye-bye.
Thank you.
Bye-bye.
Thank you.
Bye,
Bye.
everybody.
Jim,
Pleasure to meet everyone.
everybody.
Bye.
Bye.